Today I'm going to demonstrate for you the setup and the preparation and the procedure for the Catalase and Yeast Spheres lab. This is one where I'm going to demonstrate the procedure for you today, and then you'll design an experiment that will be performed later this week. So what I have in here is a mixture of yeast, which are living organisms, and they contain an enzyme called catalase, which I'll talk about in a minute. And it's mixed with a sodium alginate solution, which if you've ever had something like bubble tea, those little gelatinous bubbles, are made of this stuff. So the first thing we are going to do is we are going to drop some of that into the solution of calcium chloride. Hang on, I'm grabbing a dropper quick. When we drop this solution into calcium chloride, it will form those little bubbles that you see in bubble tea. So I am just going to make some of these so you can kind of see how this works. As we drop it in here, it's going to kind of form these little solid beads, and I'll try and kind of hold it up there so you can see them fall into the ground. There. And they solidify into those little bubbles that you get in bubble tea. I'm just going to make several of them so you can kind of see how this works. There we go. And we'll let them sit there for just a minute and solidify. The enzyme we're using in here, catalase, is an enzyme that's found in almost all living cells. And what it does is it breaks down hydrogen peroxide. And you might have heard of hydrogen peroxide. You can get it at at CVS, at the pharmacy. It's used to sterilize things. But it's also produced as a side, like a byproduct of a lot of the chemical reactions in living things, and it becomes toxic if we have too much of it. And so living things need to detoxify that, and the enzyme catalase helps to do that. So there's catalase, because there's catalase in the yeast, there is now catalase, that enzyme, trapped in these spheres. And really the only point of the spheres is to hold the catalase and to make sure we have relatively the same amount in each of our spheres. I'm going to grab a spoon and a beaker of water real quick just to rinse those spheres off, and then we're going to test this reaction so you can kind of see how it works. So I'm going to scoop these spheres out of this solution now and just put them into some tap water. And you can see they just form these nice little beads. They're kind of that rubbery texture like bubble tea. So I'm just going to take a few of them out and rinse them off a little bit so that we can use them to test this catalase reaction. So now I'm going to take this test tube. And what I have here in this graduated cylinder is 25 milliliters of a solution of hydrogen peroxide. And this is a 0.6% solution of hydrogen peroxide. I'm going to move these out of the way so that you can see what happens here. And so what happens in this reaction is as we drop these spheres into the hydrogen peroxide, at first they will just fall to the bottom of the test tube, but then when they break down the hydrogen peroxide, they break it into water and oxygen, and as that oxygen is released, the spheres will float back to the top. So let's see if this will work. So it sinks to the bottom, and as that reaction begins to occur, hopefully in just a second here, we will see our spheres starting to float, maybe. Starting to float back up to the top. Come on, sphere. I think my hydrogen peroxide might be too old. Crap. Well, that's disappointing. All this work. Let's see, we'll give it just another minute here. See if that one's gonna float. And we can test this rate of reaction by timing how long it takes. There we go. From when it hits the bottom until it hits the top. And so by doing this, so I'll do it one more time so you can see it. You can either start timing right now as it hits the surface, or you can start timing now when it hits the bottom. It really doesn't make any difference. That's just one thing that we will need to keep consistent as we do this experiment. And the longer it takes for that sphere to rise, the slower this reaction is happening. So we can use this type of setup to test some of the different factors that affect the speed of an enzymatic reaction. And tomorrow you're going to learn more about enzymes and some of those factors that can affect the speed of that reaction. Specifically in this lab, we're going to look at one of three factors. You're going to choose either the pH, the temperature, or the concentration of the hydrogen peroxide substrate. There it goes. So obviously our time for some of these would be a little bit long. So you're going to go into Google Classroom and you will see a document there that gives you the background on this lab and also asks you to design an experiment. You will have some more time tomorrow to continue working on this experiment design and ask questions, but start thinking now about how you could use this setup to design an experiment and start writing out a little bit about how you would set that up and how you would run that experiment. We will continue talking about this tomorrow. Have a great day.